So today I'm going to talk about the embedding, how the embeddings really learn, and also I'm going to introduce how the geometric analysis of the model we developed, we call it AMProCo representation look like. So before that, I'm going to introduce myself. I'm Mojtaba Masoudi. I am a research data scientist at National Oceanography Centre. I am as a part of the biological carbon cycle group at NOC. In our group, we are addressing multiple aspects of biological carbon pump, and we have expertise from imaging system, autonomous underwater vehicles, machine learning and AI, and model observation synthesis. My background is pure in computer science, and I am especially expertise in image processing and deep learning. The reason people hire me in the NOC was I was trying to leveraging AI for marine biology, and I am particularly interested in the advanced machine learning model, unsupervised and self-supervised learning. And for some project, we were also involved into the automated real-time models as well. So for the measurement and collecting the data, there are many techniques for collecting imaging from the oceans. For example, we can operating remotely vehicles, autonomous underwater vehicles, fixed platforms. We can use gliders, fixed towing samples. So a lot of things can be do the measurement for the oceans. Here is a few examples of the measuring system we apply for capturing our data, ship-based platform and autonomous platform. Currently in-house, we do have different type of the imaging systems. We have underwater vision profilers, CPICs, Least hologram imaging. So each of them targeting different size of the organisms, and obviously they do have a different modality and appearance in the imaging. In the right side, you can see the red camera frame. We use that red camera frame for deploying at the same time different imaging systems. For example, in this picture, you can see we are deploying UBP5, CPICs, and Least Hollow at the same times as a profiling. So what's the motivation for using machine learning? So machine learning helps to researcher to do the automation and speed of the analysis of the data. It helps to reduce human bias. For example, for the plankton identification, it can handle the high volume of the data with the parallel computing and the efficient algorithms. And also it can cover the new pattern and relationship on the data. And also you can benefit of the real-time monitoring with the help of the AI. But the problem we do have with the machine learning is the obtaining a very curated data set can be challenging, and sometimes I can explain sometimes it's not possible to do that. So it need to do the careful curations and the consideration for the training data, and also if you applying for specific tasks for your training, it can pose a challenging, for example, for the rare and novel species. So before I'm jumping to my research, my recent research, I'm gonna give you a quick recap about what we've done before. we published this paper in 2024. This paper was about how we use the self-supervised learning for optimizing plankton image classifications. We use the metadata enhanced representation learning to optimize our model. The model itself, the idea comes up from the idea that the distribution of the plankton data can be affected by different factors like water temperature, currents, depths, locations, different like abundance of the plankton can be informative. So what we started was we did have a label. We did have a data set without any labels. So we designed a pretest task on the unlabeled data. So what we did was we pre-trained a model with the pretest task we designed it, and then we used this representation for the transferring the knowledge for another model, which is smaller and also balanced subsets. So in that case, we create a robust feature representations, and then we did the clustering and extracting the features and supervise like a fine tuning for balanced representations. For the result, we did our model on a data set that comprised from the 980 dives between the up to 5,000 meter depths, and it expanded around the broad range of the oceanic regions. And here I'm showing how our clusterings use those feature unsupervised to cluster different pattern of the imaging. And about predictions, we in that paper we discuss how the depth can be improved the result. For example, if you have a 10 class naive data set from the 33 F score, with the depth regularization we can increase it to the 53, and even you can with a multi-view classification you can increase it to the up to 55, which was a promising result. So in this paper we did massively experiments regarding to the different parameters, like different metadata profiling, and also the different like a sampling technique and different classifying. So to do a started our idea how it started, I need to introduce you some concept in the machine learning. The first thing is the long-tailed distributions. So for anybody doesn't know about the long-tailed distribution, it means when you plot your distribution of your data, you can see the distributions histogram look like this. We do have, if you can separate it on the head and tail classes. For the head classes, you will see just a small number of classes account the majority of the occurrence, while there are a lot of other classes that only occur less frequency. So in that case, when you give into the standard classification, you will realize the problem, because the majority dominate the training, and of course you have a poor performance for the minority classes. And here is a few example of the long-tailed data sets. So after people realize the long-tailed data set can bring the problem, they started to publish the data set like a long-tailed distributed, and then you can test against your model if the model is working properly in the real scenario. So the data we have in the plankton is massively long-tailed distributed. It's, for example, if I give you a roughly, sometimes for some data set, you can have like 80% of the data only occurrence for one classes. Another things is in this paper was interesting for me because the people introduced the bias can also happening regardless of the label. For example, they have a look at the ImageNet dataset, which is a very famous dataset that constructed in 2009 through the internet search and crowdsourcing, and they realized that bias can happen, for example, in the gender, skin color, and age. And you can see how, for example, the male comparing to the females data, anything relevant to the male is more than the female in the data sets. For example, if you searching the banker, you will realize you can bring more male than the female. That can be problematic for the typical machine learning model. And also they mentioned that the bias can also happening during the cleanup as well. So annotator can give a more positive response to the stereotypes. Another thing is about the mini batch size. So to understand the mini batch, you need to know about some basic optimization in machine learning. So how the machine learning is optimized is we're giving an example to the machine, and the machine trying to optimize the answer. If the answer is wrong, is tweak the parameters of the machines, and how it's decided about the changing parameter is based on the gradient descent. So it decided which direction it needs to go. It needs to reduce the error. For reducing error, you need to go into the negative direction of the gradient. So if we wanted to go in calculating that, we need to have the whole data, but it's not possible to get in whole data sets. So we need to approximate a mini batch of example. And then the idea of the stochastic gradient comes up. And the people showing that this noisy mini batch can also helpful for the calculating the error. So with considering those things, we come up with the idea of the constructive learning. We introduce a way of the modeling in a constructive learning. So in the constructive learning, the whole idea is we pulling positive pair together and pushing negative pairs apart. So after the training, we have an augmented view of the same imaging, have the same directions, and the different imaging have a different directions. So in this concept, we mapping data in the hypersphere, in unit of the hyperspheres. And so in this type of the learning, we are instead of learning the label directly, we learn the space when the distance is really a matter. And for that one, we introduced the AMProCo, which is the adaptive multi-prototype probabilistic constructive learning. So AMProCo is using a probabilistic constructive learning. We instead of learning each samples, we learning the probability of the data, and also we show in that model the multi-prototype can be helping the training. So we use the multi-prototype learning, and also this multi-prototype is adaptive. So depending on which type of the classes we are learning, it decided adaptively how many number of prototypes it need to be assigned for each classes. So here you can see the loss function we introduced in this model. So this AMProCo is currently under review and it's going to be published soon. Regarding the experiment, we did the experiment on 2 different data sets. One on the CFAR data set. The typical CFAR is using 100 classes, but they change the CFAR to the long-tailed distributions, and we use that data set. And for UVP, of course, it's massively long-tailed. So here is the illustration of how we deal with this type of the data. We introduce the superclasses for the prototypes. So we group, for example, all type of the different taxonomy relevant to copepod as a superclass of a copepod, Nidaria, likewise, and etc. We introduce about 20 superclasses for the planktons, and similarly for the CFAR, I was using the superclasses which was already allocated for the CFAR dataset as well. In terms of the performance of the AMProCo, I showed that AMProCo can increase the performance for these 2 type of the data set. I did report on the 3 type of the classes: many shot, medium shot, and few shots. So for few shot, we only have a small number of sample per class. For example, it can be everything less than 20 samples goes to the few shots. Everything between 20 and 100 goes to the medium shot, and the classes more than 100 samples going to the many shots. So I was showing that AMProCo, the model we develop, can increase from the typical probabilistic conservative learning from 44% to the 49%. And even for the few shot models, it doubled the performance from 9% to the 18%. Here on the back, I'm showing how the multiple prototype can affect the performance. So the shaft value is introduced here based on the different type of the modality. As you can see, the green show how the fixed number of prototypes can affect the performance. So sometimes for some classes it can be helpful, but sometimes is not. And overall, you can see multiple prototype can be beneficial for most of the superclasses. Okay, so here are the things how we can use this model in reality. So we did the model development, and now we wanted to use it in the reality, in real world data we're capturing. So this comes the idea of how we use the geometric analysis. To give you an example about incredible works our group did for data capturing, we collecting a massive data from the North Atlantic oceans with a different type of the instruments. For the biocarbon data, we're collecting data from the ALR, floats, glider, CTD, and fixed platforms, and we capture around more than 5 million samples for specific locations. So what we've done, we already have a training with a reference model which comprised from the massive data set around the world. We use that model as a basic, and we transfer to another network, and we did the fine-tuning for biocarbon data. So we create a fine-tuning model So here is a summary of the overall class composition based on the different model we have. As you can see, every model have a different opinion about the classes, and the composition is more or less different from the different models. So these three type of the model is the UVP model, UVP6 model, fine tuning, and Ecotaxa, and also use the deep learning model. We use that model as well for the comparison. So how this model composition comparable with the taxonomies? We did investigations about the CTD data, because for the CTD data we do have taxonomist labels. So from the 44 deployments from 0 to the 1,000 meters, we analyze how the machine learning is comparable with the taxonomist predictions. As you can see on the left, the effective number of the AI model has the higher value. It means the model can predict more classes than the taxonomies, and for taxonomy's opinions, the model only concentrated on the fewer classes. And on the right side, you can see how the model concentration is different based on what the AI predicted and what the taxonomist has opinion. So for the taxonomies, it's more confident on a small number of classes, rather than for the AI model, which the classes is more spread out. And if you wanted to compare against the AI models, so is more or less they are having similar opinions, but you can see how, for example, how UVPNet model identify more classes for each profiling comparing to the others. Also, for example, fine-tuning model tend to concentrate on a small number of dominant classes. And on the right side, you can see the fine-tuning and the UVP6 model concentrations is more confident on classes comparing to the Ecotaxa model, which is spread out. So the question is, are these labels we have are reliable? In our team, when we have a look the data composition and asking the taxonomies to give us the summary, we realized the system is really complex to rely only on the labels. So in that case, we thinking about we do the geometric analysis of the representations. So we get started to how we can summarize our data, because we know that our data is on the unit of hyperspheres. We had a crowd of the point of the data, so we had 1,000 of the embedding per deployments, and what we try to do was we don't want to change the meaning of the distance between the embedding, and we don't want to touch the structure of the embedding. We just only wanted to capture different aspects of the geometry. So if you wanted to compare the typical classifications model with the metric learning we've done for the classifications, or the boundary is important. It's important because it doesn't look like the data. It's only trying to separating the data regardless of where it is. So the separation is matter, but for the geometry model we had, the distance is meaningful, and for example, similar sample are closer to each other, and neighborhood is important as well. So we create a similarity graph, and then I introduce some basic metrics for the analysis. The first thing was how the average vectoring is pointing. So we calculate the centroid norm. If the centroid norm was high, it means all the embedding on that deployment are aligned. If it's low, it means we have a mean mix of directions. The other metric was effective rank, and effective rank introduced how many independent directions we have. If we have a large value, it means we do have a complex geometry in our data. And also we have a look at the tail coherence. So it was important for us to see how the rare and rare and noise of the data look like. So if it's high, it means the rare samples are consistent with the rest of the data. So the general rule was we can't separate the data based on the different structure in the geometry. So we created some names for those geometries. Monodominant, structural diversity, rare events, patchy community, balance, turnover, regime mixture, highly mixed community. So it's completely depend on the metrics we calculate. For example, if the central norm is high for our data and the tail is relatively closer, it means the data is coherent. But if the effective rank is low, it means that the data structure is simple. If it's high, it means there is a complexity happening in the data as well. So here I plotted how the concentration and complexity for our deployments is different. You can see how different deployment can be varied regardless of the concentration and complexity. I plotted here how the centroid norm is comparable with the effective rank, and you can see most of the deployment having a higher effective rank complexity means more complexity, but at the same time simple community and more aligned together. Here is the plot of the 168 downcast ALR deployments. So in this plot from the beginning, how the ALR started until the beginning, how the central norm is changing from the higher at the first deployments to going down, and then it fluctuated in the middle, and then it gets relatively in the middle value for the later deployments. Here I was showing how the deployments can be vary based on the different parameters. As you can see, the central norm and cosine of the P10 similarity are relatively following the same rule, but effective can be vary. But one interesting thing was another metric we calculate about the data was how the mean centroid cosine similarity comparing to the other look like. For the last plot, you can see for most of the deployments are relatively similar to each other, but in the middle of the deployments, it reduced down. It means there is something happening in terms of the community, but after those deployments, it started again to going up and being similar to the first deployments. So how we think about, can we use this label to introduce the diversity? So we investigate how the label look like. Here I plotted how the Shannon index of the diversity of the label comparable with the central node. You can see for most of the deployment is following the same rule. It means if the central norm is high, means Shannon can be high or can be a low. If it's high Shannon, it means there are only a few labels dominant and the embedding are aligned, and we do have just only one regime deployment, mostly probably detritus. And for those have the Shannon value higher, it means we do have a lot of labels, but the embedding pointing in many directions. So it means that the community is diverse and mixed. But there is two type of the deployments in the data as well. One was when the labels says diverse, but the embeddings still look like aligned. And that purple circle deployments, that might be one dominant directions in the data, or that might be a model might get wrong about the label, model collapsing probably. And also we do have some deployments that can tell us how the, for example, label are diverse when the central norm is relatively in medium. So the embedding are not extremely aligned, but we do have a main direction of the data. So here in this plot, I show that the similar pattern of the data for the different metrics, but one is interesting for me was when we do have the Shannon diversity from the CTD, we do have a label for those as well. If when you can see here the labels prediction comparing to the Shannon of the taxonomies are relatively following the same rule, and it was an interesting and promising observation for us, and it's showing the same pattern until the very last deployment. It's fluctuated a little bit for the prediction channel, but for the taxonomies and prediction, regardless how it calculates the labels, it doesn't matter, but you can get the overall trend of the data and the deployment look like regardless of the what is their label names. What we concluded from these plots. Here I show you we did investigations about the depth profiling as well. We do have resolutions for the deployment based on the different depths bins. In the left side, I'll show you how the central norm is look like, and I coloring based on the if it's higher, it's more brighter, and if it's lower, it means the community is diverse, is not more aligned, and it's more darker in the color. And you can comparing with the taxonomist label relatively following the same patterns. When we're showing here, for example, when the dominant labels proportion is high, means only one label dominates the samples, and it can be less, can be a dark value when the number of label proportion is less than for the whole specific depths being profiling. And here we comparing the bin depth profiling comparison between the CTD data and the ALR data. We realize the homogeneity of the data from the surface until the for example, for the ALR 280 depth profiling, it's increasing. It means the homogeneity increasing, but the complexity decreased from the surface to the down. But the overall structure, it doesn't look like changing from ALR data. For the CTD, showing at the left side for more depth value for 1,000 meter data set we had. If we want to compare with the same depth value, we have that data which following exactly the same rule for the CTD as well, and we realize those CTD and ALR data, regardless how we collecting the data and the way of the data was completely different, can be comparable, and we can get some insight from those data as well. And that was my, I think, my last slide.